Hello, thank you for joining us. We're just waiting for a few more people to log on and then we'll get started. Thank you for your patience. All right, it seems like everyone's here, so I'll get started. So first of all, thank you for joining us and welcome to this webinar about teaching with cells in 3D. My name is Kulsum. I'm an application specialist for education at Nanolive. Nanolive is a company based in Switzerland which has developed a microscope that can image cells in 3D. It's called the 3D Cell Explorer. Cell.academy is the educational spin-off of this company, and that is what I'll be speaking about today. So last month, we officially launched the Cell.academy learning space. It's an interactive online platform with 3D cell data, and the idea is to add another dimension to your biology lessons. In today's webinar, I'll start off with a brief overview of the 3D Cell Explorer, the microscope that has been created at Nanolive, because all the cell images on the Cell.academy learning space are taken with this microscope. I will then provide an overview of the learning space with its contents and examples of cell data that you can find on there. I will then do a live demonstration with our interactive software so you can see exactly how it works and you can see what the students are expected to do with it. If you have any questions during the live webinar, please use the chat box on the right-hand side. It will send us an email which I'll respond to as soon as possible. Otherwise, feel free to email us at education@nanolive.ch. So there's absolutely no need to convince you as biology teachers that cell biology is fundamental to provide basic understanding of biological processes, since cells are the basic units of life. However, the main limitations of traditional methods with compound microscopes is that students have to work individually and they observe cells in low resolution and in 2D. But most of all, these samples are dead, and that's very far from a science that is supposed to study living things. That is why at Nanolive, we realize that the 3D Cell Explorer has great potential, not only for high-end research, where it's being used all over the world, but also for education. So this is the 3D Cell Explorer. It's a holotomographic microscope, and it uses the physical properties of the cell based on the refractive index of the different organelles, and that's how we construct an image. And this image is at extremely high magnification. You can get it at 1,200 times, and the resolution is 200 nanometers. So you can just imagine the power of this microscope. A great advantage is that we can observe living samples since we don't apply any staining or any fixative agents. So the image that we get is not only in 2D, but we can also get a 3D render of this and 4D time-lapse videos of dynamic processes which are stopped over time, and you can have a look at them in 3 dimensions. So it really takes learning potential one step further. The applications are not only limited to 3D renders of cells and 4D time lapses, but you can also do quantitative analysis of cell size and volume, for example, using our software. And you can then definitely observe cell dynamics and interaction between different cell types. The applications are limitless. Moreover, this is a state-of-the-art research tool. It's being used in laboratories worldwide. And this is great to encourage and prepare students for the research tools that they may be using in the future. And of course, it gets them excited about pursuing careers in science as well. So by incorporating the 3D Cell Explorer in classrooms, it provides a more collaborative approach to learning cell biology, as the students can interact with the data and work in teams. And it provides a dynamic learning environment for the students. They also relate more to digital tools, given that they grow up with them, so it's definitely more exciting for them to work with computers. So here we have an example of human cheek cells, which are observed with both types of microscopes. On the left, you can see the cells stained with methylene blue, to highlight the nucleus. This sample is quite old and it shows dead cells with minimal detail. This was taken with a traditional compound microscope. On the right-hand side, you can see a higher resolution representation of a cheek cell taken with a 3D cell explorer. And by using fresh samples right from the mouth of the students, you can see the 3D render. The colors that you see here, they added digitally with our software. So you can isolate different structures such as the nucleus in blue or the membrane in green here. It really gives you a more global representation of what a cell looks like. You can even see bacteria at the surface of the cell in red, and this is due to the high resolution of the microscope. This is always great to really give the students a concrete idea of the difference in sizes of different cell types. So here you can compare bacterial cells to a human cell. And this type of result really increases the learning potential for students. So here we have another example of onion root tip mitosis with a compound microscope. And as you can see, the resolution is quite low and out of focus due to the different cell layers. But with the 3D Cell Explorer, you can get more detailed and comprehensive images in both 2D and 3D. So on the left-hand side, you can see the raw data that comes from the microscope, which is the kind of data that you will find on the Cell Academy learning space. As you can see here, we're traveling through the different stacks of images, so in the Z plane, and you can then see the different layers of the cells and focus on the area that interests you. We can then use our software to add colors to the different components of the cell. And as you can see here on the right-hand side, we have the chromosomes in green, which you can really see here in going through anaphase. So we have various customers in Switzerland, Germany, and the US who've bought this microscope and are really excited by the prospects of this microscope. We have some testimonials, which always the same ideas come up, as you can see. It's about engagement and interest of the students for microscopy, the uniqueness of this technology, and the innovation that it brings. This really proposes new ideas for teachers to motivate the students for cell biology. So as we have seen the high interest for incorporating 3D cell data into lessons, we then developed the Cell Academy learning space, which is a more budget-friendly alternative to getting the microscope. So I will now go over what we offer and the different options that are available with this learning space. So subscription to the Cell Academy learning space gives you access to a library of cell images, all taken with the 3D Cell Explorer. We also provide detailed lesson plans and worksheets to guide both teachers and students through the exploration of 3D and 4D cell data. The subscription also gives the whole school licenses to our Cell Viewer software, with which the students can interact with the data, add colors to the cells, and work with cell size and volume measurements to complete the worksheets. We have two different offers. There's a standard package and a premium package. I'll start off by explaining the standard package. So there are 5 of our most popular lesson plans, which are all aligned with international curriculum standards. The NGSS for the United States, the National Curriculum for the UK, and IB worldwide. This covers lessons for grades 6 to 12, and we have examples of cheek cells, onion root mitosis, blood and diseases such as anemia, and carbohydrates as well, as we observe the starch granule in a green banana versus a ripe banana and understand how sugar is stored. And we have a 4D file of asexual reproduction, meaning using yeast, we have fission yeast and budding yeast, and you can compare the two types of reproduction with looking at dynamic cell time lapses. So let's take the mitosis lesson package, for example. As you can see, we have multiple raw data files, which are in grayscale. These are the files that the students will be working with. We have different stages of mitosis, prophase, metaphase, anaphase, and telophase, and we can travel through the different stacks of images to then focus on different planes. The learning goals are clearly defined. Firstly, students will identify different structures in the onion cell and distinguish the different stages of mitosis. And then they can also take it a step further and explore the role of mitosis in maintaining this complex organism And we have some exercises which are more mathematical, where they have to work with unit conversions to really understand the size of cells. If the students have done the cheek cell experiment early on, then they can also compare the similarities and differences between plant and animal cells. And all of this collects evidence for the cell theory that all living organisms are composed of cells. Moreover, if you'd like to try a demo version of our software with a sample file and a worksheet, just to have an idea of what this will actually look like, you can fill out the form on our website. All you have to do is go on cell.academy/login, and there's a form to fill out on there. As mentioned previously, we also provide 4D data files for more dynamic processes. So right now we're going to have a look at asexual reproduction. So here's a video of fission yeast. So as you can see, we can rotate this 3D render. On the left-hand side, we still have the raw data file. And at different time points, you can pause the video and rotate the image, have a look at the different planes. And you can really see how the single cell is going to divide into two through fission. So what's exciting is that then students can use the ruler tool to then measure the different sizes of the cell, and they can really explore it at their own pace and stop on the frames that interest them as well. Of course, our worksheets also guide students through this exploration. So now we can move on to our premium packages, which are slightly more extensive. So these premium packages can be supplemented to the standard plan as per your wishes. So you can tailor your lessons to your interest. They can be added on at individual purchase. So we have a vast array of cell types in both 3D and 4D. I will go over some examples now. So for example, we have cell death. As you all know, cell death, there are two types of cell death. There's necrosis, which is spontaneous, and apoptosis, which is programmed. In textbooks, you can see these two different types of cell death are very distinctive in terms of their dynamics. So necrosis is more of a swelling and disintegration of the membrane, whereas apoptosis is more of a condensing of the membrane and then fragmentation. So I like to compare it like as explosion versus implosion. So here we have a cell, a mammalian cell in culture that we added NaOH to, so sodium hydroxide, and that induces necrosis. And you see this effect is instantaneous. The cell swells, and you can really see this in the 3D render, and the inner components of the cell are just flowing out and it flattens up completely. So you really have a perfect representation, a live representation of what was previously shown in textbooks. For apoptosis, so as you can see, we can measure cell height, cell size, and cell volume with this data. For apoptosis, we just leave a melanoma cell to die at room temperature overnight. And you can see that the cell dynamics really change. So once again, the volume and the height are really changed, and this helps students to understand the process and visualize it in a more realistic way than the flat images that we're used to seeing. Here you can see as it's really become much higher and smaller, more compact. Another example of lesson plans that we have is with bacteria. So this is great for quantitative analysis. We can follow the growth of bacteria in an alginate bead and then analyze the volume of these colonies. So here we have an example at T0, 0 minutes, of two single bacteria that we can identify, and we have a volume of these two bacteria. So then if we leave the colony to grow, we can then, after 2 hours and 50 minutes, see a clump where it's hard to differentiate the number of bacteria that are in there. But because we can determine the volume with our software by simple extrapolation of of the volume of the colony divided by the volume of a single bacteria, we can then determine the number of bacteria that are in this colony. This experiment can be repeated with different colonies, different time points, and it can also be left for longer time, so overnight, where we have a huge colony of 172 bacteria. So from analyzing this data, the students can then plot a graph of the growth rate of the bacteria, for example. And this provides live evidence for the formula of growth rates, which they always see in books and mathematical exercises. So as you can see, there are endless possibilities of processes and cell types that you can observe with these different lesson packages. At Nanolive, there are scientists that are working on new applications which we constantly add to the library. So this data is all unique and it provides information that was previously inaccessible. So here we have an example of mouse embryonic stem cells that are dividing. And as you can see, when the chromosomes are condensed during mitosis, they highlight in orange. This is all added digitally with our software. And it really gives you this idea of mitosis being a continuous process and not like the distinct stages that we imagine from textbooks. Over here, each cell division takes about 20 minutes. This video is sped up. And we have the raw data file which students can stop on and work on at each time point as they wish. Now I will move on to the live demonstration of our software. So what you see here is our interactive 3D cell viewer. It is the software with which you can then load all of the files, do the 3D renders and cell measurements of size and volume. When you subscribe, you'll receive login credentials. To log into the learning space, you just have to click on the cloud icon here at the bottom left. And then you enter the credentials that we send you. And the password, and log in. So here you can filter and reach on the learning space. This is my private database, I have many other things as well. And here you go, you have all of the standard lesson plans and also the premium ones that you may have purchased. To open one, you just click, for example, on mitosis. And here you'll see a link to open the worksheet and vol files. Vol files are the 3D raw data that we provide. So these are the ones that you can open then with our software. Some of the lesson plans also have stain files. I'll show you how those work in a minute. Let's start off by opening the first vol file. So you just click on the link and it opens automatically in Steve. Here on the left you have the 3D cell data. So you just have to travel through the stacks to observe all of the different layers of the image. This is the image that has been taken directly with the microscope. You can also use the slider down here to travel through so you can see at which level you're at. That's useful for measuring cell height, for example. What you see here on the right-hand side is an automatic render of a 3D model based on an algorithm that the software uses. So it's just to have a preview of what you expect to see. If you'd like to add colors on your own, you can just switch from the color map, which is this render, to staining, where then you have full control and you can then add colors as per your wish. So here we'll start off with the chromosomes. What happens is when you color a few pixels of a certain grayscale, which is a certain refractive index, anything else which has the same refractive index will also get colored in the same color. And that's how then you build an image by separating different parts of the cell. So here you can see the chromosomes quite nicely. I will choose a color here at the bottom, let's say blue. And then you just roughly choose an area of interest. And as you can see, it highlights here on the right-hand side. You can then adjust the staining panel. You can adjust the opacity. We have some tutorials that help you get a nice staining that you can see on our website. And then that's how you can nicely build colorful images based on your preferences. What we also offer are staining files that are already pre-prepared. So for example, if you just click on stain file 1 over here, it opens up a staining file in Steve that we've already prepared. And here you can then rotate it and have a look. There we go. So you may have noticed these numbers that have appeared on these boxes. These are actually values of the volume of your staining, and that is quite useful for some other lesson plans that we have where we ask questions about the volume of certain organelles. So you can also then use the ruler tool to measure different cells. You just click on one end and another, and there you have it in micrometers. You can see 36.9 micrometers for this cell. You can then try another cell, and you can very easily build an average value from that. So we have worksheets that can help you build your lesson. So let's open the one for mitosis, for example. These are in PDF format. It provides a short summary of what to expect, which grade level this is for, the origin of the sample, and then we have the learning goals that we had covered earlier on during the presentation. And then we also provide some background information each time just to get the teachers ready for the lesson with all of the key words that may come up during the lesson plan, and some questions for engagement for the students before starting the lesson, just to get them started on the lesson and see what they already know. So just a few questions. And then we have full worksheets with some more theoretical questions and some questions requiring opening the different files on the cloud database, so on Cell.academy Learning Space. We also have different questions about measurements using the ruler tool, and there'll be some unit conversion that's needed to do. So it really covers quite a vast array of questions. And at the end, we have alignment with education standards, citing nicely which ones are being covered in this lesson plan. So there you have it. As you can see, it's a really easy tool to use. It's really intuitive, and students pick up on it very, very quickly. And that covers the content of today's webinar. Thank you for your attention. Please visit our website, cell.academy, and feel free to contact us with any questions to education@nanolive.ch, and we will respond to you as soon as possible. Goodbye.